Pleasure to introduce the first speaker, Thirumalai. He doesn't need any more introduction. So the title is Growth Mechanism of Amyloid Fibrils and Oligomers.
So Sri, thanks for, this is way up, no? Should I? Inviting me, feeding us like crazy, making sure we are unfit to do any work when we go home, and the fantastic hospitality to everyone. So it's been fun. We'll be back. The talks yesterday, especially on, the whole colloid day was very fascinating. In particular, the problems that are posed for hot sphere crystallization, seemingly simple, apparently still being worked over, and we don't know exactly what's going on in terms of reconciling, from what I understand, experiments and computer simulations. And the mechanism is still, as far as I could figure out, not fully sorted out. The interesting thing about that problem was, once you state that problem, everyone in this room would say, yes, it's something that one ought to think about. It's sufficiently simple that you encountered these in some form or the other in graduate school or elsewhere. But the subject matter that I'm going to talk about today, and it probably holds good for most of the day, the problem may not be as well defined, and in particular, the precise statement of the problem will depend upon who you ask. Let me give two completely contrasting scenarios for the relevance of protein aggregation. What is protein aggregation? The impetus to work on this problem largely comes from the linkage between these protein aggregation and what I call now deposition diseases, of which there are a number. And in particular, there is an increasing awareness that we probably ought to think about slowing down the process by which these neurodegenerative diseases kill people, most notably, say, the Alzheimer's disease, which has been one that's been on the market for a long time. The big pharmaceutical companies in the United States spends about $1.5 billion per drug from the time you conceive of a potential drug till it becomes available in the market. So the people who are really interested in trying to figure out how A-beta aggregation is linked to these diseases are not really interested in knowing the kinds of questions that you would be interested in, namely how hot spheres crystallize, or analog of that problem in protein aggregation. The kinds of things that they worry about, and I'll give a little bit of introduction to that, although it's not germane to the thing that I want to tell you about, because you guys are interested in different kinds of questions. So I'll try to make this talk a little bit closer to what you might want to think about. These problems are sufficiently complicated. They involve an interplay of chemistry, biochemistry, metabolic processes, and of course, probably computations and bioinformatics. And the role of theory is not entirely clear where it in fact fits in. On the other extreme, there is a growing number of people who have been trained in quantitative science, especially in physics, say, who want to get into these problems. And one of the first things they want to know about is, are there universal principles that are involved in dictating the aggregation of proteins? And I think if it goes beyond the universality, one typically, or most physics-trained scientists typically lose interest, especially if their training has been in physics for a very long time. So I'm going to tell you this joke, which paraphrases this sort of attitude towards biological problems, and I don't blame them, because there's a sufficiently large number of interesting problems in physics that you can sort of work on it the rest of your life. This joke was told to me first by Adrian Parsegian, who is now in UMass, used to be in NIH, and in fact has appeared in Physics Today at some point, say 10 years ago or something like that. So it turns out a physicist is tired of condensed matter physics problems, say, and he wants to know how brain actually functions. And he has a neurosurgeon friend, and he goes and tells him, listen, I want to switch fields. I want to work on how brain actually works. and tell me something about it. So this guy, the neurosurgeon, says, well, let me start from the beginning. The brain has an inside and outside as far as signals are concerned. And the physicist says, stop, stop, you have told me too much already. And so that's the other extreme of the problem. I'm going to hit some middle ground here, although my interests are with time going more towards understanding some of the basic problems involved in the production of A beta and things like that. But that's something, I think it's a little bit too complicated, and we haven't digested this ourselves, and I won't talk about that. So before I get to the talk, this work that I'm going to tell you about has been done by a terrific postdoc. In fact, he actually wants to return to India, preferably to Hyderabad, where his father has a farm, apparently. And I don't think he wants to do farming, but he wants to be close by. And John Straub, who is in Boston University, and the work has been funded for about over 10 years by NIH. So let's think about the fate of a polypeptide chain when it's actually made. It's not really in a cellular context. So we know that the ribosome actually makes proteins after reading the messenger RNA. This is a very complicated, regulated process for which 3 people won the Nobel Prize last year for determining its structure. And I must parenthetically add, even though the structure is in fact known, the mechanism by which the peptide synthesis is made is still not entirely clear. Very often, after the protein is actually made, it's protected by all kinds of chaperones depending upon the systems that you're looking at. And then there are sometimes post-translational modifications that are involved. And in fact, one such post-translational modification is what produces the A beta peptide. If there is no post-translational modification, then you have an unfolded protein in a crowded environment, and it's usually, of course, finds its way to a folded state, which is a requirement for it to be biologically viable. And after the function, it's degraded by an proteasome machinery, which is, again, in fact, extremely complicated. Occasionally, there could be accumulation of unfolded proteins. People speculate, for example, the late onset Alzheimer's disease could well be because your chaperones don't do the clearance very well, and therefore you then occupy some state, which I'll keep referring to N star, and I'll describe this in detail. And these could in fact form oligomers, which then subsequently can grow by multiple stages into amyloid fibrils. The language that one uses from this part of the till the end of the reaction is very similar to the kinds of language we heard yesterday. There's nucleation, there's delay time, and then it rapidly grows, and one wants to understand the nature of nucleus and things like that. There are no even starting point for writing down what ought to be the driving forces and the forces that oppose this in any simple analytic form, like the homogeneous nucleation theory that we heard about yesterday, to which apparently you still have to add terms, and in fact those terms could in fact be relevant here, and I'll probably make a comment. That kind of terms could be relevant here as well. And that's a fate of a typical polypeptide chain. Most of the time nothing goes wrong, but sometimes, in fact, things can go wrong. The problem of protein aggregation and formation of oligomers, in fact, was dormant for an extraordinarily long time, until possibly in the early 90s, or maybe end of the 80s, when it was recognized that there are many, many post-evolutionary diseases, which are sometimes called deposition diseases, conformational diseases, and so on and so forth, that come about because proteins don't do what they're supposed to do, fold, function, get cleared, and then process starts itself in the regulatory processes. There are many more diseases than the ones listed here. AD is the most, one of the most famous ones, and it's linked to the aggregation of a well-defined polypeptide chain, which is just 40 or 42 amino acid residues, and its aggregation, or the oligomer form of that is in fact related to the disease. And then of course this mad cow disease, which goes under the name of transmissible spongiform encephalopathies, and there are lots of them, became famous, and they're linked to the aggregation of prion protein. People who take dialysis, like Osama bin Laden, for example, in fact, may also have to watch out for some kind of aggregation process, and this is a big problem in Japan, and that's associated with aggregation of, I think, beta 2 microglobulin. So the thing that I think has interested a lot of physicists, at least a few of them are in this audience, like Sri and Muthukumar and others, is that they look at the end structure of this reaction, this complicated reaction involving the transition from monomers to full-grown fibrils, and the fibrils are characteristic structures, and I'll show you a little bit of that, and then one might just conclude that there ought to be some underlying universal things that one can learn about it. Maybe, maybe not. Our current thinking on this problem, which I will not discuss at all, is really related to this particular slide in the A-beta case, which is associated with Alzheimer's disease. A-beta is a product, cleavage product in a metabolic processing of so-called amyloid precursor protein, which is a membrane protein. It's embedded in this membrane region. And if you blow this up, this is sort of the sequence that you get. There are multiple mutations that occur. For example, there's something called the Iowa mutant, Flemish mutant, Dutch mutant, et cetera, which differ in the amino acid at one single site, and this, in fact, alters the growth kinetics of the fibril itself. The reason the big pharma is extremely excited about AD, especially this year, there have been 3 news articles in Chemical Engineering News, is because this scission of this APP that actually produces A-beta protein is coordinated by 3 membrane proteins, which are very complicated, which are called the secretases, alpha, beta, and gamma. In fact, the scissors means it cleaves at this particular site, and depending upon exactly where the cleavage actually occurs within the membrane, you'll get a variable length A-beta protein. It could be from 39 to 43, but typically it's not range. A-beta of 1 to 42, it turns out, is aggregates at a much faster rate under conditions which are realizable both in vitro and in vivo. So we are actually mostly thinking now about how the secretases actually perform this cleavage reaction and what are the structures that can accumulate, because the belief is that if you sort of knew what the membranous structure of this APP is when it's interacting with, say, the gamma secretase, which is the drug target of most of the big pharmaceutical companies, then you might be able to think about methods by which you can slow this down, if not prevent it. So this is the kind of structure that you get. This is the membrane, and this is the APP. It's really helical. This is the gamma site, and there are lots of complications associated with it. But once the scission occurs, what you go back to is the problem of production of the A-beta under in vitro conditions, and then you really try to find out how that in vitro aggregation process occurs, and that's where most of the progress has been made. So I'll tell you about a toy model, which is really very much in the line of thinking of physicists, I would say. And then I'll tell you a little bit about a fragment of the A-beta protein, and then another fragment about SUP35. It is quite clear, unlike in the case of the crystallization of hot spheres that we heard about yesterday, where there were many reasons given as to why there's a discrepancy between experiments and computers, one of them being that the solvent, in fact, Dave White's talked about that, but here it turns out the solvent plays a very critical role, perhaps both in the polymorphic nature of the fibril itself and also in creating a barrier to quote-unquote nucleation. So that's the sort of thing that we're thinking about, but this is what I'm going to tell you about. The first question that I'm going to address is the following. Is all the fluctuations in the spectrum, the free energy spectrum, the free energy folding landscape of a monomer, have any information about the kinds of structures that you're likely to get in the final structure? So is it worth studying the monomer itself? Then I'll go to this growth of monomers and make some speculative remarks about nucleation. I'll then more or less show that what we have found so far through mostly computations cannot be fully correct. And then I'll tell you about the role of water, which I think is extremely fascinating. Because these disease-related proteins are intimately related to folding, it's only the misfolding, in fact, that gives rise to potential aggregation if it hangs around long enough under the cellular cycle, it's useful to study how you want to think about how monomer proteins fold. And Jayant Udgankar, who is sitting in the audience, has done a lot of very pretty work on this particular subject that you'll hear in an hour, in half hour or so. There's a lot of theology associated with how, in fact, one imagines proteins actually fold, and it depends on which school you go to as to how you want to think about it. But generically, people would say that if a protein is quote-unquote foldable, which means there's nothing really incorrect about it, and you could think of many, many proteins under in vitro conditions that would fold, self-assemble reversibly many times. SH3 domain, for example, is one. There are lots of proteins like that. Then if you start from the folding trajectory from anywhere in this multidimensional landscape with near unit probability on a very short time scale of the order of, say, some tens of milliseconds maybe or more, depending upon the length and the sequence, etc., you will get to the Nehru basin of attraction, and that's really the desirable point. Then it can function and get cleared as this complicated diagram initially showed. Occasionally, about in E. coli, it turns out about 5% of the proteins actually don't do that. And these 5% of the proteins then require molecular chaperones. And then usually they get stuck in this competing basins of attraction for arbitrarily long times. And unless you have a chaperone machinery that's well evolved, as is indeed the case, not only in prokaryotes but also in eukaryotes, then they are rescued, and most of the time things are in fact all right. If there is a breakdown in the chaperoning machinery, then and some other clearance mechanisms that are not really working efficiently, which you might expect is the case as you get older, and then some problems can occur, and then the population of molecules in this competing basin of attraction with exposed hydrophobic residues, etc., can be sufficiently large that it can trigger aggregation. That's why looking at monomer folding is actually important. In fact, once you learn about this protein folding in whatever way we want to think about, you can immediately come up with scenarios for how aggregation might actually occur. And this is a diagram from an article that I wrote about 7 years ago. And there are several scenarios you can think about for aggregation. The particular case could be that the molecule is actually folded, which means it's ready to function, do its function, but there are some denaturation stress or something in the cell, and that occasionally occupies the so-called N-star state, which I'll call the aggregation prone state, and for a long enough time so aggregation can proceed from here. So in this scenario, this molecule has to partially unfold to occupy this metastable intermediate, if you will, and then with sufficient concentration, you can see aggregation. In the A-beta peptides, which I'm talking about, they are completely unfolded, at least to the extent that one can actually think about. There's no persistent structure. In that case, the unfolded molecule will have to traverse by collision or otherwise into, again, these metastable intermediates, and then it can fold, perhaps through helical intermediates, and finally to cross-beta structures. We know from lots of very beautiful work from Jeff Kelly for the case of TTR, which transthyretin, which is linked to some cardiomyopathy problems, that this belongs to this scenario, and you can correlate the fibril production rate to the rate of unfolding from N to N star. The case of the mammalian prions that I'm not going to discuss here, we think occurs very, very differently. We think that the mammalian prions, and it's an exception, we think that the folded state itself is metastable with respect to something that's aggregation prone, but the barrier separating between the two is sufficiently large that during the clearance process, these are never in fact populated, and you don't have to worry about it. We were lucky to make some key predictions in 2002 and 2004, which now seem to be at least quasi-accepted. So this is how I'm going to develop a toy model. This lovely structure. So one of the things people want to know about is what are the structures of the fibrils, and there is no unique structure for the fibril. We now know that, unlike folded states, where the structures are in fact unique. This is a structure of A beta 140, determined. It's a structural model determined by my good friend Rob Tico and his company at NIH. This is one form of the fibril, and what you see is a double layer structure. So this is one single layer, and the fibril axis is running away towards you, if you wish, through this axis. And then there's another layer which is just on top of it, and this particular one has a C2V type symmetry, and this is one polymorphic A beta 140 that you can realize in the laboratory. The first 9 or so residues are disordered, and the point that you should remember is it's got a strand and a bend on another strand. Each monomer is in that conformation, and they in fact stack in parallel as you go towards the board or away from the board, and then you have 2 of these forming the unit structure. The strand's axis, beta turn is perpendicular to the fibril axis, and then if you take the X-ray diffraction of this, you'll get the characteristic cross beta structures there for this and for many, many other proteins which form fibrils. Now if you look at the this is what I was talking about. This is a strand. There's a bend here. It's stabilized by this salt bridge between lysine and this D23, and this is another hydrophobic residue, and this is the structural motif of this in the monomer state. Okay. So the monomer of this A beta in the fibril state has this beta strand turn beta strand motif. It's disordered in isolation. If you want, you can think of this as a very low concentration. We then looked at these structures, and this is a toy model that we made about 10 years ago, and over time we dribbled a few papers out, and it's really not something that the bio guys care about, but this toy model is sufficiently interesting. In fact, most of the programming for this toy model you can do on this computer that you're using to read your morning newspaper. So the question is the fibril structure encoded in the monomer spectrum? And you can answer this question extremely precisely with this toy model. The toy model has 4 types of monomers. I will simply say they're called hydrophobic, polar, and plus and minus. Of course, they have no bearing to real amino acid residues. There are only 8 beads in this, 8 residues, 8 beads in this chain per monomer. So the number of sequences is just 4 to the 8. And this is the universe of amylomes, if you will, the set of sequences which can in fact be amyloidogenic, and I'll tell you what that means. The number of conformations, self-avoiding conformation, on a cubic lattice for this 8-mer is only 1,841, which means at any temperature, you can in fact enumerate everything about the system, calculate the partition function, specific heat, whatever you want, determine the folding temperature. This is a spectrum of energy levels of this monomer for one particular sequence, and the native state or the ground state is as compact as it can be on a cubic lattice. And then there are degenerate states a little bit above in energy and some above here as well. So I'm going to focus on this n star because I know it'll come up, and if I calculate the probability of occupying this n star as a function of temperature for these various parameter values of interaction energies, which are completely irrelevant as far as you're concerned, as far as I'm concerned, and you see it has a maximum. If you tune the hydrophobic residue, that's what 0 means, then the probability of occupation goes down at some appropriate value of the charge interaction. You can actually maximize this n star population. What you could do, this is another sequence where the n star in fact lies just above, is the first excited state, it's force-fold degenerate, but these are all like n star because they can topologically be trivially turned into this in this structure. If I now do a computer simulation, this is no longer an exact enumeration, it's a computer simulation. You put this in a box, the specific concentration, and these computer simulations, while easy to do, are still time consuming, so that we only do things like 10 monomers and 16 monomers. If the number of monomers is 10, you get a single layer in which you have a structure which corresponds to n star that's completely stacked and close packed, and this is the lowest energy structure that you can in fact show by repeatedly doing your computer simulations. As you increase the number of monomers, it goes in this double layer structure, again with this n star being the fundamental building unit, and this is like a hairpin, and these hairpins are stacked exactly in parallel in this case, like the A beta that I showed you, and there's a transition from a single to double layer as the number of monomers in fact increases. So these are some of the characteristics that you actually see of the fibril structure that I showed you for the A beta 140, but please do not take this to be literal. It is meant to simply give you an impression that in fact one can fool around with this toy model if you wish. So lots of things you can do with this toy model that you cannot do in experiments so easily, and one of them is what is the interplay between the interaction energies between the charged residues and the hydrophobic residues. And this is for the so-called wild type sequence that I showed you. And if you change the interactions, if you keep the hydrophobic interaction constant and change this plus minus a little bit, you get again a fibril structure, but this fibril structure, the hairpins in fact alternate as you go across the row. This is going like this, up and down, this goes down and up, and so on and so forth. It's still a fibril, and this is a polymorphic fibril for a slightly different interaction energy. And then this is a double layer structure, and if you go to some other value of the E plus minus, you get a completely compact structure, but there's no fibril at all as far as you can recognize, but therefore this wouldn't qualify as a fibril. Now, because it's a simple enough model, but not super simple that you can easily do this on desktop, but although I think you can if you wait for a couple of months or something, you can calculate the fibril rate fibril formation rate in some Monte Carlo units, which is irrelevant, as a function of temperature, and you find these characteristic curves which are seen in experiments. There's an optimal growth temperature at which the fibril rate is the maximum, and just to give you a feel for the time scale, the growth rate for the fibril is about 4 to about 9 or even more orders of magnitude greater than the time scale in which a monomer would actually fold in the absence of interactions with the peptides. So fibril takes much longer to fold, and you could design sequences in this where this n can be 15, 20, and so on and so forth, but that's very difficult for us to follow, so we don't care about them. You can also show the seeding, which means that if I take a small piece of this quote-unquote fibril or protofilament, and you put it in an otherwise sea of non-interacting monomers, in fact, at speeds of rate of formation, all these are semi-consistent with experiments, although there are no quantitative comparisons because this is, after all, a silly little model. But this is a point that seems to really be established now reasonably well, both through the work that I'm showing you here, but also through some more serious simulations and experiments from David Eisenberg, that there is probably a correlation between the rate at which fibrils actually form, that's tau fibril, logarithm of that, and the extent to which the so-called N-star state is actually populated. N-star state is that conformation in the folding landscape of the monomer, which in fact adopts a fibril-like structure when you go there, and it's by no means unique, especially when you come to realistic models. There's an ensemble of structures that can produce fibrils of differing morphology. So this is, again, this is sort of interesting to those who are interested in this. There's a logarithm, if you plot the logarithm of the fibril rate as a function of the population of at which this P n star is a maximum, that in fact is linear, and that means it tells you that it is not possible, or it's not very easy, as people thought, that you could talk about the universe of sequences that adopt fibril structures by just merely looking at sequence alone, i.e. bioinformatics alone may not be sufficient. You really have to at least worry about the monomer spectrum. It suggests, though, that if I could really thoroughly study the monomer fluctuations of folding landscape very, very carefully, either by experiments or computer simulations, it could give you the kind of structures that you can actually pack to give different kinds of morphologies, and hence it's kind of useful. So monomer landscape encodes the structure and possibly the growth rate. Okay, this is, again, to impress you that I've sort of read a couple of papers in physics, that if you, in fact, you can do some growth rate measurements as a function of the number of monomers, and that for reasons that I don't want to get into, it follows the Lifshitz-Slezau growth law, which Lifshitz, in fact, announced in this magnificent paper about 50 years ago. Okay, this I don't want to talk about. So now I want to make the same point, but now for a realistic model, and this is the TICO hairpin structure, beta turn hairpin in the fibril, and this is a structure that was produced by Reddy by looking merely at the monomers 1 to 40, and by looking at the folding landscape of this entire monomer, and occasionally you would find a high free energy conformation in which this D23K28 salt bridge that cements the VGS turn forms, and this is in fact the ensemble of N-star structures. So it is indeed the case, just as the toy model suggested, that these conformations that you find are in nascent form, already present in the folding landscape of the monomers. And this is a very thorough study by Bogdan, who was a graduate student of John Straub, and this is the spectrum of free energy states, and the D23K28 with the K28 solvated is one of the high free energy states. In the ground state, in fact, it's a completely random coil, and in order to access this high free energy conformation, there's a barrier, and then if you hang around long enough, in fact, you can nucleate this to form the A beta structures. But there are also some other higher free energy structures which have salt bridges buried, which conformations are different, and we speculate, in fact, that the polymorphism encoded is also encoded in the spectrum of N star, whereas in the lattice model, the N star is quite unique. Here, there's an ensemble of these structures. So that's the first thing that I want to tell you about. So in order to make this point absolutely clear, so a corollary of this is that if I preform this salt bridge in the monomer, which you can in fact chemically do, you would predict that the growth rate will be considerably greater than in the wild type, where this salt bridge is in fact absent. And that experiment was done by before our calculations by Steve Meredith and Rob Tico. They showed that if you preform that salt bridge, the barrier is reduced, and then there's a rate enhancement of the fibril growth by about by 1,000, and that's related to the desolvation, the necessity to hydrate the waters on the salt bridge to actually form this, which does not take place spontaneously in the monomeric state. Okay. So now I want to talk a little bit about this particular set of reactions for A beta peptide, small peptide. How does it go from unfolded state to oligomer? Part of the motivation why people are interested in oligomers, there are two reasons primarily. One, the oligomers are pretty difficult to catch experimentally. Certainly, the transition kinetics associated with them are not easy to measure. The structures are even more difficult to measure, although there are some interesting developments this year on A beta, where the oligomer structures are beginning to appear and some constraints. And the second reason is there's a current, maybe it's said in the next, no. There's the current hypothesis in the AD, the Alzheimer's disease field, due to Selko, is that it's oligomers that in fact are neurotoxic, with the fibril being the end product. And the reason these guys know this may be the case is because they inject mouse, for example, with A beta oligomers, and there's a loss of potentiation, which can be recovered when the soluble oligomers are processed through the cell cycle. They recover their memory, et cetera, et cetera. So they think the oligomers are important, although I don't really worry too much about this. So let me spend a little bit of time telling you about growth of oligomers. And we've done a number of these peptides, and they're all interesting in many, many different ways, but I'll just focus on one of them, which is a 1622, and its sequence will appear in a second, I think. And we want to know how it goes from monomer to oligomer, and initially we thought that if we really sat on this in terms of a computer simulation for a very long time and did very careful measurements, you would even be able to tell what is the critical nucleus sizes, what are the heterogeneities involved in the nucleus, et cetera, but ultimately we had to reduce to some semi-analytic arguments to in fact make that conjecture. So the computations are done using molecular dynamics, but this time we have water in it, because we really want to know what water is actually doing to this aggregation process, and then you sit and do these simulations carefully. This is the kind of computations I'm going to report to you. And this is published already about 3 years ago in PNAS. So you start with a random coil and a preformed oligomer. So M will be minimally 2 or 3 here. So you have a dimer. And then you follow this reaction, and this reaction, of course, can go back and forth. And you do this for n equals, we did this till 6, I think. And then simulations are almost impossible to carry out later. I think I've said this before, this deposition disease are neurotoxic with the plaques being the end product, etc., etc. So you preform this, have a random coil, it then adds to this, everybody fluctuates, and you see what the end product is, and then you analyze this with whatever methods you want. So this is the sequence that I showed you before. This the sequence that I'm going to talk about now is this sequence KLVFFAE, and this is in the interacting the lysine here interacts with the alpha secretase, and there are important mutations in the so-called central hydrophobic cluster. The Iowa and the Flemish and the Dutch mutant are all in this region, and therefore people in fact are focused on this quite a bit. And it's also important in importing stability to, oops. So the oligomer assembly. So insert this and then do MD simulations. This is like a confirmation, if you will, from a simulation box. These blue guys are waters, and you notice that these strands are arranged in an anti-parallel fashion, and in the monomeric state, they're random coils. The strand content in the monomer is about 1% or so, and therefore completely negligible. And this strand conformation only occurs when there is interaction that drives the assembly of the A-beta 1622. So this is a terrible slide, especially in the 21st century. Even I cannot see this here. We just want to show that, in fact, that these are anti-parallel. And so if you take the dot product of the cosine between the two, it's jumping quite spontaneously between -1 to +1. And you can read this in a paper that we wrote about in 2003. And this is anti-parallel. That it should be anti-parallel here is relatively obvious because the K and E are charged and the middle is hydrophobic. And so if you put the hydrophobes together and the charges on the opposite, you both enhance the formation, enhance hydrophobic contact, plus satisfy the Sol bridges, and that's why this is anti-parallel. Incidentally, I should say, it is quite unusual to find anti-parallel fibrils. Most of the structures that I know of, starting with A-beta, and SUP35, URI2P, and so on and so forth, they're all parallel sheet structures. Monomeric, negligible alpha content. So this is an energy landscape, if you will, in terms of some principal component analysis. And the point for, I think it's a trimer, and is there are many basins, in fact, that are occupied. The lowest free energy structure here is indeed an anti-parallel structure. And if you want, you should think of this as a little pneumatic droplet, and these are fluctuations associated with the pneumatic droplet. The point is, the soluble oligomers, they're not intact like the fibrils. They, in fact, can dissociate, reform, adopt random structures, and to the extent that these are in fact connected to the loss of potentiation in mice, it suggests that AD is not really is not linked to a regular structure like would be the case in the fibrils, but in fact something that's fluctuating. So then we wanted to talk a little bit about whether in fact you can get something about nucleation here, and so we started. We couldn't do the simulations long enough, and so in order to characterize these oligomers, soluble oligomers, we in fact computed a liquid crystal order parameter and a potential of mean force in this order parameter. So we're looking at this as the slowest mode in the system, and compute the potential mean force in that, and see if in fact one can actually use that in some case. Before I show you some of those arguments, let me tell you that these in order to accommodate this new soluble monomer without structure, and if I look at the fluctuations in P2 cos theta of these preformed oligomers, they fluctuate one hell of a lot. Those of you can't see it from the back. It goes from 0.8 to sometimes pretty close to 0. That means when I insert A-beta 1622 in a random coil state, in order to accommodate this, the original structure that you have undergoes large-scale fluctuations, and after it assembles and P2 cos theta finally goes to 0.8, corresponding to the A-beta 6-mer, you still see fluctuations, but largely only in the directionality, but not in the conformations. So the conformational fluctuations, the number of really distinct basins that you have keeps decreasing as the size of the oligomer increases, and this of course is what you expect, because once it crosses something that I'll call a critical nucleus, it must in fact form a protofibril whose fluctuations has got to be small. Coming back to water, the interior of this little pneumatic droplet is pretty much dry. Because it's a computer simulation, we can actually measure anything you want, pair correlation functions, orientational order, etc., etc. Here is a residue, 16 to 22 of one monomer and another monomer. And if you look at the contacts between them, there is just no water in there, and there's most vividly seen, there's no hydrogen bonds in here as well. And in fact, we know, and we showed this some time ago, but you can look at the movies of this, that the initial drying process in which water is expelled from the solvated monomer occurs relatively early. And it's a subsequent rearrangement of the side chain motions, plausible 1 or 2 hydrogen bond that stabilizes the monomer, the oligomeric structure. That's a rate limiting step. So in this particular sequence, which is largely hydrophobic, water is not important except in the earliest stages it's expelled. And I'll show you the next case. This is not the case. We showed, in fact, if you look at the acquisition, the initial beta content of this solvated monomer is pretty close to 0. At the end, when it's fully accommodated, its beta sheet content is of the order of unity, which is the value that's commensurate with the pneumatic droplet itself. So you could in fact ask, how does the acquisition of this beta content increase for the solvated monomer as a function of time? And that's in fact what is shown here. It's beta as a function of t. By diffusive process, it initially docks onto this fluctuating pneumatic droplet, and that happens very quickly. On that time scale, the beta in fact increases from nearly 0, say, to about 0.4, 0.5, and then there's a very slow rearrangement process, which is not yet complete for this 6-mer. It's still going on. The final state should be about 0.8, and this is still around 0.75. We stopped the simulation somewhere here, and so there are at least two distinct timescales that are involved in the locking and the docking of this monomer to this fluctuating pneumatic droplet. And one knows by just measuring this that the locking time scale, the second time scale in which bulk of the acquisition, perhaps the formation of the hydrogen bonds, side chain contacts, and so on and so forth, that's a rate limiting step. It's much greater, typically 100 to 1,000, and we can't be sure, but experiments tell us this in fact is the case. This is sequence dependent. I'll come back to that. So now this is our attempt to sort of estimate what ought to be the critical nucleus for this little problem. And one way we thought we should do this was, if you cross a critical nucleus, then the time scale in which an added monomer would dock onto this underlying fluctuating pneumatic droplet should then become independent of the size of the growing pneumatic droplet. So it'll be purely diffusion limited. So then we thought we could pin this down, but it turns out that 6 was not sufficient for us to do. So we went back and said, let's think about this P2 cos theta as the reaction coordinate, which probably isn't. And then we computed a potential and mean force. Once you compute a potential and mean force, in that degree of freedom, oops. I think I'm going to leave this here. Basically computed the, sorry about that, I apologize. We computed the diffusion coefficient in this order parameter reaction coordinate, and using that and Cramer's theory, essentially, we could compute when this would be diffusion limited, i.e. the barrier would be irrelevant, and that turns out in our estimate to be of the order of 8. So we think that size of the critical nucleus for A beta 1622 is probably 8, a little bit larger than that, although for some other systems, like for example PRPC that Giant is interested in, it's we suspect it's 2. So we don't really know if it's correct. So you might naively think that I should be able to go back and write down some sort of free energies like you do in the homogeneous nucleation theory, but it's not very obvious, because if I look at the free energy difference per monomer between, say, the monomeric state and the fibril state, it's not very large, and there's lots of cancellation going on, so it's not super clear. One. Secondly, as Dave was pointing out yesterday, these fibrils are by no means unique, and certainly the oligomers are by no means unique, and therefore there's entropy associated with that, and it's very hard to compute that in this particular case, even in an ansatz sense. So the picture of the oligomer growth, which is something that has been proposed by us and also Sue Lindquist, is that this bunch of fluctuating monomers form this disordered oligomers, if you will. They order themselves, and this goes back and forth until you reach some critical nuclear size in which the fluctuations are still there, but it's not as large to drive it to this disordered oligomers, and the new ones are incorporated by a template type mechanism that we think that's easier to understand. You could in fact put some numbers in here as well in order to measure this free energy barrier. Let me say that it is extremely unlikely that in these processes there's a single unique free energy barrier. There's going to be a distribution of free energy barriers which is really indicative of the heterogeneous processes that actually govern the formation, at least up until the protofilament stage, if not later. So when I say delta G dagger, you really should think of this as an average delta G dagger, although that's really more or less meaningless. But if you go to very large fibrils, and then you ask what is the time scale in which the monomers would add to these fibrils, you could in fact say that the fibrils are essentially stationary, and then you can use Smolakowsky theory for association, including this Debye correction, if you will, and these are all somewhat standard. We can derive this for concentration dependent of viscosity. That's a new thing we did. And then if you look at this growth rate, it is linear in concentration at small c, and that's what you would expect. This is and moderate values, it has this dependence. These two have been experimentally seen, and a very large concentration should decay as 1 over c. So the proposal is that if you could make very careful measurements, incidentally these measurements are not easy to make. You should just ask Giant, who is here, because when you vary concentrations, lots of crazy things happen to these proteins, and you cannot be completely sure about what's going on. So it's easy to write these things down, but eventually you may not be able to really get good numbers until you can in fact do very precise measurements. But if you could, or a very wide range of concentration, at least you can get some idea what the average barrier height is. It basically slows down because what needs to happen is that at very large concentrations, you're crowded by many peptides, and then you have to encounter this fibril. There's a probability of that, and that actually slows down the process. Okay, this is the last thing I'm going to say, and if I'm cut off, that's perfectly fine. But this is a completely different mechanism for assembly, and this is a peptide that comes from the E prions. N-terminal domain of the E prions. It's also a small 7-mer. So because I'm more or less will run out, let me show you what this is. There are 50 peptides for which X-ray structures actually known, all coming from David Eisenberg. And the models that I showed you were NMR models based on solid state NMR. This is the crystal structure of the so-called SUP35. This is an all-polar sequence. And this is quite important even in Huntington's, although in yeast this does no harm. If you look at the structure of this, there are a few things that I want to point out. The sheets in the strands, incidentally, this also in isolation would be completely a random coil. But in this fibril, you have this, it forms a beta strand. When you're looking at the beta strand arrangement in one sheet, they are arranged completely parallel. And this is another sheet in which they're parallel. The fibril axis runs perpendicular to the arrangement of the sheets, but the sheets themselves, the strands on the sheets, are anti-parallel. That's one thing, but this is again a cross beta structure. In the crystal structure that Eisenberg determined, he imagined that the sheets would be parallel, like two parallel sheets, and that's not the case, and I'll make a comment about that. The most spectacular thing about this structure is the interface between this strand and this strand on one sheet, and the interface between the two sheets is completely bone dry. There's just no water at all. And the side chains coming from NNQQNY across the sheets are fully interdigitated. It's a very beautiful structure and stabilized by hydrogen bonds. And so you have a dry droplet of this protofilament, although it's infinite, of course, in this direction and this direction. Okay. If you take this sheet of some finite size, about 8 to 16, and put it in water, the first thing it does is it acquires a helical twist. And this we know is in fact correct, because if you look at EM microscopes of all these fibrils, it does an overall helical twist to the fibrils. So that's what we find. But the thing I want to tell you quickly is, how does this sheet in the fibril state grow? So in order to do this, we took the sheet, removed one of the strands from the top. So if you look at it from top, there's a terrace. You added a solvated monomer, and then we follow the reaction till it goes to the completely ordered state. So that's the molecular dynamics simulation. To me, this is still quite interesting result. I mean, this appeared last year in PNAS, and this is all work of Reddy. And what is being plotted here is the growth of this beta strand content of this added monomer, which is negligible beta sheet content, as a function of time for 3 trajectories. And of course, you expect that there should be a distribution of first passage times, and hence the time scale in which different beta strand goes to unity varies. But what is really important is that the growth of the beta strand content occurs in a very stepwise manner. It's pretty close, it's very small in the beginning, and when the first passage time is reached, in fact, the jump time is extremely small. If you calculate the jump time of this, it's about 1% or 2% of the first passage time for every one of these trajectories. And once, in fact, it locks onto this structure, the beta strand remains beta content remains 1.0, which is the value it has commensurate with the lattice itself. So it's really a stepwise acquisition. I've never seen this in any other system, in fact, that we have looked at or anybody else has seen. If you look at the dynamics of ordering at the residue level, it's completely heterogeneous. And this is the distance between, say, two center of mass. And if the blue and the red coincide, that means that the added strand has acquired the beta sheet or beta equal to unity. So if you look at the various strand conformations, here, for example, this is ordered, here this is ordered, but there's no logical reason, no sequential order in which the various strands, in fact, lock on to form this very dry interface. And this is another, so I had to cite my friend's work here, Sachi, and they have seen something similar to what is called backtracking. And because I'm running out of time, I'm gonna skip some of this. And this is what happens if there's a residue, for example, that correctly forms. It has to, in fact, unfold and reform. And the point is, there's a lot of heterogeneity at assembly level. Let me go to the water part here. I'm almost done, right? I'm ready. Take 5 minutes. Okay. So maybe I'll. So now this is another, I think, well, interesting result. Shall we say? I was going to say something else, but anyway, if you look at the number of water molecules at the interface between the two strands as a function of time, the eventual state is it's got to be dry. There's no water in there. That's what the crystal structure actually says, and that's what we ought to find if this is correct. So the drying, in fact, occurs in 2 steps. As soon as the top strand finds some contact with the terrace-like structure, there's a drop in the number of water molecules. Again, everything is stepwise. It happens quickly from about 5 to 2 water molecules. And then when the beta strand goes to unity, i.e. the structure is commensurate with the lattice, the number of water molecules goes to 0, and it remains dry completely after that. This is for this polar sequence. And this loss of water is dynamically cooperative, and it's 2 stages. Stage 1 is associated with peptide dehydration of beta strand. And so I think I want to contrast that with A beta. No, maybe not. I think if you now look at the corresponding graph for this A beta case, which is driven largely by hydrophobic interactions, unlike this beta T as a function of T, which is like a step function, here you see there are lots of fluctuations. Although drying is an initial event that occurs rapidly, in about tens or so nanoseconds, whereas this drying could happen in about 400, 500, even larger number. In some cases, we never saw it at all happen, and that's what I'm going to actually show you, hopefully. Okay. So this is a different kind of simulation, and this is under review in PNAS now. So this is what will happen if I take two such sheets and let them grow. So you separate them by some distance, and it's filled with water, and then eventually it forms a dry interface. And there are parallel routes in which water is actually ejected from the hole. One way is it turns out that as you bring these two sheets together, these are polar sheets, remember, these are not hydrophobic sheets. It forms this beautiful one-dimensional water wire, and that's what you see, one water wire per sheet. And then these water wires have to be expelled before interdigitation and dry interface can actually form. And that happens either the water actually leaks out from the bottom, or it can leak out from the side because the sides are also porous. And I think this is a movie. If it works, for Steve it did not work, I think. So you can see that the water in this case is escaping from one side, and so it's showing the bulk, whereas we also have movies, which I didn't find on my computer, for leakage from the sides. And the fact that it forms this metastable structure means that the time scale of association of this all-polar sequence to form the protofilament, it has to be considerably larger than from the hydrophobic sequence. That is indeed the case. For the hydrophobic peptide, you can grow this in a couple of hours. For these polar peptides, you got to wait for a couple of days. And this is news from David Eisenberg. And yeah, this is the A beta case. You see, it never becomes totally dry. There are lots of water molecules, even though the final structure is in fact dry. There is an analogy to this A beta to this plate growth. I think I'm going to stop. I'm not going to discuss that. So let me conclude. The things that I think I've touched on are the things in blue. Electrostatic and hydrophobic interactions determine the structure and kinetics. Although I showed you most of those things in the context of the lattice model, we've shown this also in this other stuff, and people now know this to be the case. You can make point mutations and in fact screw up both the structure, enhance or decrease the kinetics in these experiments, and a limited cases have been done. The conformational heterogeneity in N star, which is the ensemble of structures that the monomer actually samples when it's not interacting with any of the monomer, seems to encode the kinds of fibrils that you actually get, both the oligomer and the fibril morphology, including the possibility that water is in the fibrils and hence has the displays a polymorphism. The growth of oligomers follows by these multi-stage dock-log mechanisms, and we can estimate some amount of it. And water, in fact, plays a very critical role independent of the sequence, but in a completely different way depending upon the sequence, both in the assembly of oligomers and possibly in creating nucleation barriers in the protofilament and fibril formation. And it is really sequence dependent. So water is really like a chameleon. Depending upon where you put it, its characteristic changes. It alters interactions in different ways. In some cases, you find this beautiful one-dimensional water wires, very reminiscent of water wires that you find in aquaporin channels, which processes water in our body, and whereas in other cases it merely acts as a lubricant that gets out of the way until drying actually happens. So there's lots of things that one can do from a biophysical point of view, although our own attention is moving towards the role of secretases of APP in the membrane context. Thanks.
Thank you, Dave.
The question was, which water model have I used for simulations? It's a very good question, and I'm a practitioner. And so we used about 3 different water models to make sure these results are semi-robust. 3 different water models, 2 different force fields, and 2 different ensembles, and to get these results.
Okay, were they like the 3 center water models, or the? It's
SPCE, TIP3P, and TIP5P, TIP4P are the ones we used.
And so, what was the temperature of your simulation?
300.
I would like to ask, don't you think there is a role of ions like zinc and copper in the aggregation?
Excellent question. Of course, this is not a course on A beta. Copper and zinc are intimately related to the aggregation process, And one ought to think about this. They were not included in this process at all. It is true that you can, in fact, enhance growth rates, and this is a very, very important point to actually think about, especially as you progress towards trying to understand the linkage to diseases.
Thank you.
I think from what you characterize as the physicist perspective, I guess one of the questions that one would like to have an answer to is why there is the apparent universality of the morphology of the aggregates, right? In other words, whenever you do have ordered aggregates, why are they ordered as fibrils? And I saw little bits and pieces of the answers, but can you summarize what is the current state of understanding of why this is the case?
I'm going to give you a nuanced answer to that question. First of all, it is true that when you globally look at it, these fibrils have this characteristic cross-beta structure. But when you examine them in detail, of course, there are changes, and these changes are not irrelevant. These changes are, in fact, linked to the virulence of the disease itself. That's point number one. Point number two is morphologies also contain water molecules. Now, if you ask the question, what should be the ground state energy, and this is the kind of physics-y type of thing that you might be interested in, of these sequences which are amyloidogenic. One of the things I forgot to mention is that these universe of amylomes, not every sequence, in fact, can be made to form fibrils on some time scale that you're looking at it. They may at the end of the day, but not when you're looking at it. But those who do, if you ask the question, what's the ground state of those, it is really the sheet that's, in fact, the lowest in energy. Just by a little bit, incidentally. It's not much. And if you take all the interactions into account, subtract and add them, sheet wins over a little bit. How generic is it is still not clear for these peptide sequences. That certainly is the case, and one can work this out through some of these simulations and also through the crystal structures from David.
Possibly related question.
Yep.
In terms of nucleation, the scenario that you talked about,
yeah.
I guess if you just think in terms of nucleation of a one-dimensional growth,
yeah.
right? There is no barrier. No, I mean, it should be spontaneous growth.
Yeah,
but I guess the complication or the added feature here is that you need to fluctuate out of
exactly.
the native structure.
Right. Yeah.
And that's where the barrier comes in.
The barrier comes in two places. It comes prior also to the photofibril stage. After that, it's just adding monomer. You can think of that as a quasi one-dimensional growth.
Statics matters, or its ability to exclude a hydrophobic solute matters. So do you think the multiple roles that water plays in this process have to do with different aspects of water as a solvent? I mean, whether it's around a polar residue or around...
Absolutely. That's absolutely the case. That's why I didn't show this long enough, because it's a bit more easy to understand, namely the association between the hydrophobic plates, which happens in picosecond timescales when you look at doing the computer simulations, and it's really the expulsion of the water around this hydrophobic area, just like in the A-beta case. Whereas in the polar sequence, the water, even though it's losing its hydrogen bonds with the bulk, it's very happy to establish new ones with the asparagines and glutamines, and hence you form this lovely wire which are mesoscopic, which coherently flip, and so on and so forth. So that's what I meant by context.